Morning, everyone. Thanks for inviting me to talk today. StemBANCC is a large consortium which the name of is based on this rather tortured acronym here, stem cells for biological assays of novel drugs and predictive toxicology. Now, I've changed this talk 3 times, so I apologise if there's not enough science in it, and I apologise if there's too much science in it. So I didn't know how to quite strike the balance. So the project wouldn't have been possible really without the discoveries of John Gurdon and Shinya Yamanaka on reprogramming factors which can turn an adult cell from an individual to an induced pluripotent cell. And then you can take these cells and potentially differentiate them into any other cell type in the body. You can model diseases with these cells. You can use them for screening and for drug toxicology. So the StemBANCC consortium is based really on this ability to make iPS cells from any individual and potentially turn these cells into any cell type in the body. Some other discoveries just before StemBANCC got going were these papers here, where people had taken cells, mainly fibroblasts, from individuals with different genetically inherited diseases, and made iPS cells from them, and then turned them into the relevant cell type. For example, from Ludovic Vallier's lab, he made iPS cells from various liver-associated diseases and then turned these cells into hepatocytes. So today I just wanted to give you an overview of StemBANCC. It's a huge project, so I'll have to go relatively quickly, and then I'll say a bit more about Work Package 10, which is the Cell Differentiation and Toxicology Work Package, which I lead together with my colleague at Roche. So the idea of StemBANCC is to take 500 patients or healthy individuals, take fibroblasts from these. This was the original plan, although we've altered this now, and we also take blood in some instances to reprogram. It was decided early on to use the Sendai virus system, so the reprogramming factors aren't integrated. And the plan is to make 1,500 iPS lines. So this would be 3 clones from each individual. It makes me tired just saying that number. And then to differentiate these cells into different cell types, model disease, and use them for drug screening. And the disease areas which StemBANCC covers are diabetes, diseases of the peripheral nervous system, the central nervous system, and also some conditions that are linked to toxicology, such as drug-induced liver injury. So StemBANCC is an innovative medicine initiative, an IMI EFPIA initiative. It's a consortium of 10 pharmaceutical companies, now 11. Originally AstraZeneca were supposed to be involved, but with the reorganisation they stood down from that. They've now rejoined, so it's 11 pharmaceutical companies and 25 academic or SME enterprises. The start date was autumn 2012. It lasts for 5 years, so we're about 3/5 of the way through. The total cost was about 56 million euros, and the project coordinator is Martin Graf at Roche, and the managing entity is Sam Kader at Oxford University. It was actually them that made me come and talk today. So this is just a list of all the, I won't go through them, you'll be pleased to know. So this is just a list just showing how many different individual companies and academic centres are involved. So there are 12 work packages in StemBANCC. The first one is the management and administration. We have subject recruitment. There's a reprogramming work package, a biobanking and distribution work package. Molecular profiling, so this is patient and iPS profiling, including all the omics. So they're actually doing exome sequencing on all the individuals who are recruited to make iPS cells. Also data management, which when you realise that they're doing exome sequencing them all, you need a lot of data management. And then there are different work packages for the different areas. There's diabetes, peripheral nervous system, central nervous system, and the one I'm involved mainly with is cell differentiation and toxicology. There's also an assay development work package and also a communication and dissemination of information work package. And these are the subjects which we wanted to recruit in the beginning. As you can see, the consortium is very neuro-oriented. So there's patients with various neuropathies, Alzheimer's, Parkinson's, autism, schizophrenia, bipolar disorder, migraine, and then tacked on the end are diabetes and drug metabolism, and also a group of healthy volunteers as well. So the status at the moment is that all the important SOPs are in place for biopsy, iPS generation, cultivation of iPS, etc., etc. And the patient recruitment forms, all the ethics are in place. Some of this happened quite late because it was so neuro-oriented, they kind of neglected to set up a recruitment centre for the toxicology work package. So we've had to organise that in Edinburgh. So that's just recently started, so we've started recruiting patients with DILI, for example, in Edinburgh. The patient recruitment is almost complete. I think we're heading towards 400 out of the 500 so at the moment, with a similar number of reprogrammings underway. As I mentioned, Edinburgh has been set up as a recruitment centre for drug-induced liver injury. Also the cardiology conditions such as LQT, Brugada, and also a group of Alport syndrome, which nobody knew where to put, so it's in the toxicology work package. And this is just the procedure that we hope to follow. Some of these have fallen slightly behind, some are ahead of the game. But as you can see here, we're exome sequencing the 500 patients, and then there's going to be omics profiling of at least 200 of the cell lines. So it's a huge undertaking. And the key challenges to be addressed really are to reprogram the primary cells with minimal off-target effects, producing enough of differentiated cells for medium to high throughput screening, to try and get well-defined, meaningful disease groups for iPS generation. And we really need useful assays with decent endpoints to characterize the lines that we've made. And possibly one of the more tricky areas is to have consistent and standardized protocols to achieve fully differentiated cells. Now, the definition of a fully differentiated cells is a somewhat controversial area, and I'll talk more about this when I talk about hepatocytes and differentiating those cells. The other area which is really complete is to establish a robust ethical and research governance framework to enable future industry academic collaborations. And this shouldn't really be underestimated because it took a year to sort out all the ethics and all the different competing interests before we could actually start the project. So we started off almost a year behind. So I just wanted to talk more about work package 10, which is the work package I'm most familiar with. And here we hope to generate functionally mature target cells of toxicological interest from the human-induced pluripotent cell lines that we've made. And we want to do this in a robust and scalable manner. And in Work Package 10, we're trying to develop protocols for renal cells, both proximal tubular cells and podocytes, brain aggregates. Although there's individual cell types being made in the other work packages, this is designed to try and make an amalgam of different brain types. The other two more well-established protocols are for cardiomyocytes and also for hepatocytes. And we also have a component of toxicity pathway analysis which links into Work Package 5. As I mentioned, myself and Nicole Clement at Roche are the co-leads in Work Package 10. So this was part of the original plan was to, for the hepatocytes at least, was to, and also the cardiomyocytes, was to take 100 of the cell lines and run them through the established robust protocol. However, it's become clear that, if you ask this question, are stem cell-derived hepatocytes sufficiently mature to use them mechanistically and or predictively? The answer is maybe. So I could stand and echo much of what Catherine said yesterday, but we're singing from the same hymn sheet, really, that we can get the cells so far, but not quite to a fully mature state. And the assays that we use now are really based on what we've been doing since the 80s, late 90s into the 2000s, really, looking at adult hepatocytes, which we used to perfuse bits of liver, get the adult hepatocytes, and do various studies. So all the ELISAs and PCR, etc., were in place. And these are some of the studies. This is a Mac to PowerPoint glitch here, but some of the things that we use to characterize the cells include hepatic export proteins by ELISA, the expression of various membrane transporters, cytochrome P450 function, expression of membrane transporters, and ureogenesis. So the problems with the current hepatocyte protocols are that once they're fully differentiated from iPS cells, as far as we can take them at the moment, they're very like freshly isolated adult hepatocytes. They die pretty quickly, within a few days. Even on Matrigel, they may last a week to a week and a half. So they have a very short life in culture. So one of the key things is to try and improve the longevity of these cells once they're differentiated. The other question is, do differentiated hepatocytes achieve mature function? Clearly they don't. They don't achieve mature function. They probably get to a kind of fetal hepatoblast stage. So one of the key things to try and do is also to improve the function of these cells. So in Work Package 10, this is just a few graphs to illustrate what we know so far. So this is the kind of standard protocol which was established in Edinburgh in collaboration with David Hay and his group. And we've done quite extensive microarray analysis looking at these differentiated iPS cells in comparison with, if you like, real primary adult hepatocytes. So this is a very simple example here. This is albumin. So you can see that the cells, as they differentiate through the different stages, never quite get to the same production or gene expression of albumin. And this is very similar for all the other things we've looked at, CYPs, different acute phase proteins, different export proteins. So they never quite get to what an adult hepatocyte can produce or metabolize. So this is a problem. So we've looked at maybe 20 different iPS cell lines generated by StemBANCC, and there is great variability on how good they are at differentiating to a hepatocyte. And this variability extends even to clones from the same individual. And just as a small example here, this is clones from SBAD3. So from one individual, this is clone 4 and clone 1. And you can see here, this is the export of fibronectin. So you can see that clone 1 here is much better at producing fibronectin. However, if you look at the production of fibrinogen, you can see that the other clone, clone 4, is much better at exporting this protein. So there's great variability between the cell lines and even between clones from the same individual. Whether this is some residue of epigenetic imprinting, which is altering their ability to differentiate into hepatocytes, we don't know. We've also tried a small cocktail of drugs, mainly because people were arguing for ages about which drug you should use, and I think Mick Dilley's spent much of its progress doing that and assembling a large panel of drugs which you can use to assess the toxicological impact on hepatocytes. So we decided, we went with Janssen's small cocktail assembly that they use, which are these: S-bifenatoin, diclofenac, midazolam, and dextromethorphan. And we put these in as a cocktail to see if we can induce CYP activity. And these can be assessed fairly simply with the P450 glow kits from Promega. And some of the questions we're still arguing about is, are there specific toxicological endpoints and through points that we should use for stem cell-derived hepatocytes? But I think that the clearest message is that we really need to improve the maturation of the cells and also the longevity of these cells. And some clues to how we might do this have come from other studies in my lab, where we've been looking at human liver development and isolating various cell types from the developing liver, including hepatoblasts. So one of the things we do know is that co-culture with other cell types can improve the function of these cells, and that's been shown by other groups as well, for example, endothelial cells. But there's also the possibility to add in Kupffer cells and stellate cells, for example. And we know even the supernatant from endothelial cells or non-parenchymal cells put onto these hepatocytes will improve their function to some extent. Another difference between the iPS differentiation protocol and what's happening naturally during development is that these bipotential hepatoblasts, if you take them into culture, they will continue to divide. Whereas if you stop the iPS differentiation protocol at this hepatoblast stage, they just die. So we know cell interactions may be important in providing maturation of hepatocytes, and we have some evidence that placental factors and growth factors from the placenta may influence maturation and division of these cells. So we now have good evidence that hepatocyte endothelial culture, at the very least, improves hepatocyte function, and we have good evidence that placenta-derived factors are important. And we know a couple of these factors. So our key activity at the moment, rather than just bashing through 100 IPS lines using the standard protocol, is now really to concentrate on improving the maturation and the longevity of the cells when we get them to this hepatocyte-like stage. So some of the things that we're involved in at the moment are co-culture approaches, for example with endothelial cells. And there are now very good protocols for making endothelial cells from iPS cells and also making macrophages from iPS cells. So potentially we can make these cells from the same iPS line, put them together. We've also been using novel substrates and matrices, for example polymers, and we've done some work with Dave Hayes' group on different polymers which show a beneficial effect in hepatocyte function. There are also survival factors and small molecules that we've identified, so that the trick is going to be getting the right combination of matrices and factors to try and improve maturation and longevity at the end of the hepatocyte differentiation protocol. So that's what I wanted to say about hepatocytes, and I'll just mention the other cell types we're involved with in work package 10. Oh, sorry, I've just insulted Berlin and Biberach because they're involved in growing the hepatocytes in bioreactors, and you can see there is an improvement in function. This is just a simple albumin export assay, and you can see that albumin is much better in the 3D reactors than in 2D cultures. And we have comparative studies underway between the different centres to look at comparing 2D, 3D and microspheroid culture in terms of hepatocyte function. So I just wanted in one slide just to mention cardiomyocytes. So this, again, is a well-established, robust protocol, but again, the cardiomyocytes aren't quite mature when you get them. But unlike hepatocytes, they don't die immediately. They can survive in culture for a long time, and they gradually mature. And one of the big issues here is to scale up enough cardiomyocytes to provide for the different studies that people want to do. But this is progressing very well. With the renal lineages, this is more of a startup operation. So Paul Jennings' group in Innsbruck and also Jamie Davis' group in Edinburgh have been working to generate protocols to produce podocytes and renal epithelial cells. And this is progressing quite well, so we'll soon have established protocols for producing both podocytes and proximal tubular cells. We're also making good progress with producing brain aggregates, and they can now produce aggregates with all the markers for neuronal cells, for astrocytes and oligodendrocytes. So all that's needed here is to optimize the differentiation protocol. As I mentioned, the big recruiting hole in StemBANCC was the Work Package 10 patient recruitment. So we've now set up in Edinburgh as a recruitment centre, and I've managed to bully some of the cardiologists and some of my hepatology chums into recruiting patients for us. So here we'll be recruiting LQT 1, 2 and 3, Brugada patients, also Alport syndrome, and drug-induced liver injury patients. Just finished there. This is a picture just of the steering committee and the StemBANCC team leaders back in October 2012, and nobody's jumped off a bridge yet. So that's good. Thank you very much.